And we're going to move on to our next speaker, Dr. Pobbati. Dr. Pobbati is a molecular biologist at the Cleveland Clinic and co-project leader at the Cleveland Clinic Center for Therapeutics Discovery. Dr. Pobbati is credited for identifying and validating TEAD as a druggable target in the Hippo signaling pathway. Nice to have you, Dr. Pobbati, and look forward to your presentation.
Are you able to hear me? Are you able to see my screen? I put it on a high.
Yes and yes.
Yes, cool. All right. Thanks very much for inviting me to present here, particularly a big thank you to Denise for having the trust. And Hugh Leonard captured the right word. So there is significant momentum that's happening here. And I'm really excited to be part of that when there is enough momentum that is gathered in EHE research, particularly from the clinical point of view. There are so many EHE clinical trials that are happening. So by the way, like as David introduced, I'm Ajaybabu. I'm in Rubin Lab and also an assistant professor at Cleveland Clinic Lerner College of Medicine. I'd like to reiterate that EHE is caused by a chromosomal translocation that abnormally joins two genes, and the two genes that are joined, like as several people mentioned, are TAS and CAMTA1. So just want to tell you in a naive way what these two genes endow. So what this endows is monster-like properties. So the ability to turn on the genes, the TC or TAS CAMTA1 gets it from the TAS part, and the ability to stay in the nucleus, it gets it from the CAMTA1 part. So combining these properties, what it makes is like a monster. This is a minotaur that I give you an analogy of how it can combine human and bull-like properties. Although it's a monster, this monster cannot work alone. Dr. Rubin touched upon this. So it needs a partner, and this partner is called as TEAD. So the TEAD research has garnered much traction. This is not because TEAD is involved in EHE, but TEAD is involved in several other solid tumors. And because there is a greater interest of it being involved in other solid tumors, it has gained significant traction. But TAS CAMTA1 is specific for EHE community, so there is not much research done on TAS CAMTA1, and only the people in the EHE do research on that. So the wild type component of the TAS CAMTA1 are YAP and TAS. So my talk is going to be divided into two parts. The first part, I'm going to talk about targeting TEAD, and the second part, I'm going to talk about targeting TAS CAMTA1 or TC. So currently, all the clinical trials that involve TEAD inhibitors use TEAD inhibitors as monotherapy agent because or in other words, it means that's the only inhibitor that is being used. However, the preclinical work suggests that there's probably a need for a combination therapy because of probable resistant mechanism. Before I touch upon what I am planning to do or what our research is, here are the 4 clinical trials candidates that involve TEAD inhibitors, and as from the 3 of them are sponsors of this conference. So 3 TEAD inhibitors, the Vivace, the Echina, and the SpringWorks, they target what is called as the central pocket. Vivace was the first to bring TEAD inhibitors to clinical trials, and their phase 1 data on human is released. So it's really good to know that inhibiting TEAD is safe because you never know whether inhibiting that target give rise to some unwanted safety issues. In this scenario, the company that stands out is Novartis because it targets the TEAD surface. And we were really also excited about targeting the TEAD surface. And I'm going to talk briefly about what it is. And Novartis was the first to do the impossible and an inspiration for us. So the first part of my talk is called as pocket science because what we are trying to do is targeting a TEAD pocket. I differentiate myself from rocket science. So this is the analogy. TAS CAMTA1 needs to bind to TEAD. So if you put a small molecule there, TAS CAMTA1 no longer binds to TEAD. That's the whole rationale. So throughout this talk, I'm going to give you a high-level overview because of bearing in mind that this is primarily for patients. It's about empowering patients. So what happens when you put a small molecule that in the TEAD pocket is that TASCAMTA1 no longer interacts with TEAD. And what happens is that you turn off that oncogenic switch. So TASCAMTA1 cannot do its job. And this is not an easy endeavor. It requires a lot of effort. So this is collectively done in collaboration with the C3TD. The applicant PI is Sean Stauffer. So I and Bin serve as co-project leader here. Of course, our big collaborator for biology is Dr. Rubin. I'm in his lab. So this is what we are trying to target. I talk about pocket science, but if you look at the molecular level, there are actually 3 ways to target or design a small molecule that binds to TEAD. So the Vivace, Echina, and Springworks are doing the central pocket. The Cleveland Clinic, we are interested to target the interface 2 pocket, and also we are interested to target the interface 3 pocket. So the Novartis has targeted the interface 3 pocket. So this, we want to do it because this is like a direct interaction. This is where the TASCAMTA1 or YAP and TAS binds. So you prevent direct binding of TASCAMTA1 to TEAD, and that was the rationale for it. So to devise a new chemical matter, we embarked on 2 different approaches. One is called as a high throughput screen, and the other one is called as a fragment screen. And what we do in a high throughput screen, imagine this like a TEAD pocket. What we are looking for is a small molecule or a compound that binds to this TEAD pocket. So we are trying to identify a small molecule that could bind to the entire TEAD pocket. And finding such molecule is like finding a needle in a haystack. And that's why we have to screen a lot of small molecules. And we call this as a high throughput screen because it involves a lot of compounds for screening. Another approach, we do it in parallel, is called as a fragment-based screen. Fundamentally, this approach differs from the high throughput screen in that we are not looking for a small molecule that occupies the entire TEAD pocket, but actually a small portion of the TEAD pocket. Once we identified the small molecule fragment, what we do is that we grow the small molecule so that it occupies the another pocket, another region in the same pocket. So the advantage of that is that you don't need to screen a large chemical library, and relatively the fragments-based screen in drug discovery terms tend to give ligands of high ligand efficiency. So we did this through 2 methods, NMR-based screen and HTS screen. So the fragment-based screen, we use this machine. This is called as the NMR. And for a high-throughput screen, we use this machine, which is a plate reader. And both these methodologies converge at a point where we use structure-based drug design. What we do is that we solve structures of TEAD in complex with these small molecules, and then use structure as a guide to make more potent small molecules. Once we have these small molecules, in our model system, we almost always use EHE cells because it's a genetically monogenic cancer. We use the mouse model, cell line derived from it, to understand TEAD engagement. Wherever that is highlighted is where I pitch in. So I'm a jack of few trades. So I pitch in for this. So the current status where we are at is that the fragment screen in our hands has been very successful because we identified novel fragments that occupy the interface 2 and interface 3 pockets. So this kind of fragments has not been reported at all in the literature. So we are really excited about it. And what we are doing is similar to what I've highlighted before. We are growing that fragment to improve the potency. At this point, I like to tell you, unfortunately, even though we screened about 200,000 compounds, the high throughput screen gave 0 hits. I mean, like I said, it's like finding a needle in a haystack. So maybe we have to increase our library size to identify through hits. Nevertheless, we are happy to get something to move on, and which is and thanks to the fragment-based screening approach. So the second part of the talk, what I'm going to do is about targeting TC. So if you want to target TC, you want to locate TC. How do we locate TC? In our cell line, as Dr. Rubin mentioned, we have TASCAMTA1 expressing mouse cell lines that generate ESE. You can inject those cell lines and it generates tumors that resemble human EHE. That's a big advantage we have. So we detect TC through immunofluorescence, where we are detecting TC in the cells, and you see how happy TC is in the nucleus. And I know this is a nucleus because I'm staining for DNA using a dye called HIST. So one way to locate TC is detecting TC in cells via an immunofluorescence. Another way to detect TC is to detect in a blot. Here you have to lyse the cells, and you see TC is predominantly localized in the chromatin. With this in hand, with this tool in hand, we hypothesize that Well, if somehow you move TC from the nucleus, remember that is where TC's job is. That's where it turns on the gene. You move it from the nucleus to the cytoplasm, then you make it less oncogenic. So that was the rationale for this screen. But before we embarked on the screen, we did do our due diligence, because these are known YAPTAZ inhibitors. These compounds are known to inhibit YAPTAZ, and they do this thing for YAPTAZ, which we wanted to do, which is move YAP and TAZ from the nucleus to the cytoplasm. Does it do the same for TC? The answer is no. As you can see, predominantly, even after adding these drugs, which are known to translocate YAPTAZ from the nucleus to the cytoplasm, TC still remains in the nucleus. So this sets the stage for us to do a screen, because we have no clue what compounds can do the stuff that we expected to do. So we embarked on it. We devised a screen, actually carefully devised a screen using a chemical probe library. What it does is that it specifically inhibits certain proteins. And interestingly, we also used FDA-approved drugs. So the idea is that if we identify some FDA-approved drugs, that means if that can do this job, the idea is that we can probably push it to phase 2 clinical trials, because these are already approved drugs. A big thank you to the EHE Foundation. I received the grant in 2022 and also 2023 for this project. I'm forever grateful, because this is the first funding agency that funded me as a PI. And as you can see, this screen has been incredibly successful. I didn't expect this to be this successful. So we screened about 4,000 compounds. Only 1% of the compounds can move the TAZ-CAMTA1 from the nucleus to the cytoplasm. And we know that by an increase in the cytoplasmic to nucleus ratio. All of these is done by computer. It gives me the value, and then I look at the hits. And 5 compounds we have highlighted here, and that does the job more efficiently. And more interestingly, we are particularly interested with dinacyclib. So dinacyclib is what we call as a cyclin-dependent kinase inhibitor. It is an FDA-approved drug, and it seems to do the job that we wanted to do. Actually, don't want to go into the details. It's a really exciting screen, because all these 4, although the targets could be slightly different, they are all part of a particular molecular complex called as a transcriptional complex. So we are actually targeting we are actually targeting different proteins in a same complex. So here is what you will see. So TC is predominantly localized in the nucleus. When you add these drugs, CGP or DINA, one thing that you would observe is that the level of TC is decreased. Another thing that you might observe is that it's become more diffuse. You no longer see these dots. That means TC is more translocated from the nucleus to the cytoplasm, and also the total level of TC has decreased. If you don't believe me, I'm showing the same thing in an immunoblot. So upon adding DINA, what happens is that the TC actually disappears from the cell. And we know it disappears through proteasome inhibition, because when you add something called as a proteasomal inhibitor, you can restore TC level. For example, add CGP, add DINA, TC levels decrease, and then you add MG132, you restore TC levels. And this is not all CDK inhibitors do it. For example, palbociclib cannot do it. This is, for example, this inhibitor, the palbociclib, it's actually a 4/6 inhibitor that is used in other sarcoma. Recently, Bill Tapp published a manuscript about the utility of palbociclib in other sarcomas. But for EHE, it is a different cancer. It has a different mechanism of action. So dinacyclib seem to work much better. So what happens? So as you know, this is an oncogenic protein. What happens if you remove that from the cell? Our hypothesis was that the cell will lose viability, and it may kill the cells, because you're removing the monster from the cell through the administration of the drug, which is dinacyclib. And that is exactly what you see here. So the viability, as if you see this curve growing down, that means you're comparing the viability of the cells are no longer viable. When compared to 100% is when the cells are not seeing the drug. So as you titrate it with CGP and DINA, you're seeing a reduction in the viability. When we compared this with MGH-CP1, at that time, that was the known TEAD inhibitor that was available. that is about a 2 orders of magnitude more potent compared to the MGH CP1. So these drugs are working really well. And another interesting part, so this has been observed in literature also, like the TEAD inhibitors are actually not killing cancer cells. And that's what we also see in MGH CP1. What it does, at least in the cell line point of view, is that it stops them from growing, but they're not actually killing them. Whereas CGP and DINA are actually killing them. We know that because there is a pronounced increase in apoptosis or cell killing upon addition of this drug. So what this happened is that the grant that is funded by a EHE foundation set the stage for a little larger grant, because when we tell to NIH that we want to do a screening, almost always they don't like it. But when we say we already did the screen, and that's where the funding from EHE Foundation was very important. And then they say, we identified a compound and now we want to gain mechanistic insights into it. They love it. So this has been awarded to Dr. Rubin and me, R21 grant, and we are happy for that. So this is what we observed in cell line models. How does DINA fare in mouse models? So we inject mouse cells, and these mouse cells metastasis to the lung. So this is the primary tumor. You also see EHE in the vasculature here. But what more we see is that in this model system that we have, so the EHE spreads to the mouse, or metastasis to the lung, mouse lung. And when you treat it with dinacyclib, there is a significant reduction in lung metastasis. And this has been quantified here. So I reached my 15-minute time point. I just summarized 2 things here. First part of my talk, the aim of that talk is to develop a novel small molecule, and that is targeting TEAD. The second part of the talk is to try to repurpose an already available drug for EHE treatment, and that involves targeting TC. None of these has been possible without a solid team, and I'm really fortunate to be part of this TEAD project team, also part of the phenotypic screen team. So this is a combination between 2 departments, cancer biology and C3TD. And I thank you for your attention.
Thank you so much for that update. It amazes me how much progress is being made on a year-to-year basis. This is just an incredible opportunity going forward here. And along those lines, with DINA, and I believe that's a Merck drug, normally someone finds a drug candidate and then they've got 5 to 8 years of $1 billion FDA trial to deal with. When you're repurposing drugs, we, in theory, don't have to do that. Obviously, there can be FDA trials to get the permission to say it's useful for EHE, but we run into these situations where if the drug's approved for something else, clinicians can use it, what's called off-label. What do you see as the path in your research to provide enough evidence that clinicians might look at this as a drug they might use with EHE patients? So
because it's a patient talk, I didn't really delve into the mechanism of action in there. I feel we should not jump the gun, because the thing is that we identified, so DINA, the problem with DINA is that it actually inhibits, I can elaborate a bit more of mechanism of action. It inhibits 4 cyclin dependent kinase, 1, 2, 4, and 9, CDK1, CDK2, CDK4, and CDK9. So we've teased apart the mechanism of what is really important. And it's not 1, it's not 2, it's not 4. It's actually the CDK9 inhibition that is more important for its activity. So having known that, we know now that CDK9 inhibitors are already available now. This is what we want to fine tune. Why do you want to inhibit other cyclin-dependent kinases, which play housekeeping roles in the cell, which have no role in TC activity? So we may want to use a more focused drug, which is CDK9, which also does the job of inhibiting, of removing TC from the cells. So that the idea is that you are increasing the therapeutic window, so that you're not targeting, so you reduce toxicity by not targeting a housekeeping gene in there. And actually, that's kind of what I set the stage for the next grant funding. We're planning to use this approach for EHE and also probably for an R01 to try to tease apart like specific inhibitors, because DINA is targeting other CDKs. We want to focus on what is more relevant using more focused approach so that you improve the therapeutic window.
Excellent. Well, you've opened up a new direction, which looks very, very exciting here. Thank you very much. And